in the cells. Okay? So the citric acid cycle, so ang issue muna dito is the energy availability of the components of the metabolism. So we have already studied regarding the glycolysis, pero this is only the first stage of the glucose oxidation. So within the glucose oxidation, first glucose oxidation nga, glycolysis, we are creating pyruvate. So yung pyruvate, nag-release siya ng energy with negative 146 kilojoules per mole. This is a large amount of energy, and maliit lang ito it will if it will be the only source of energy for the cell. So kailangan pa mag-undergo ng different oxidation, and with the full oxidation that offers yung cell, this glucose can release a negative 2,840 kilojoules per mole using the oxidation of UCTCA and other oxidation. So, mahalaga na makapag-obtain tayo ng mataas na energy para patuloy na mag-work yung cells, and para makapag-produce pa ng different metabolites or different components, proteins, nucleic acids, or other metabolites para mabuhay yung cells pa and mag-generate pa siya. Okay, wait lang. Stop ko lang yung classmates. Alright. So this happens in cellular respiration, yung Krebs cycle. Si citric acid actually marami siyang tawag. Si TCA, si CAC, si Krebs cycle. So malin doon, that's all the same. But they are all a cyclic way of generating energy and usage of the starting material pyruvate. Okay, this of course in mitochondria in eukaryotes. This process in which cells consumes oxygen and produces carbon dioxide. It provides more energy ATP from glucose than from glucose than glycolysis. So mas marami na po-produce na ATP sa Krebs cycle indirectly and directly compared to your glycolysis. Okay? It also captures energy stored in lipids and amino acid. It is used by animals, plants, and many microorganisms to produce energy at dahil nga maraming energy na gagamit sa kanya, it is well used. So in this case, in glycolysis natin, we have already studied it. You produce 2 ATPs here. While sa Krebs cycles naman, indirectly, dahil meron siyang isang component that is directly connected with another energy producing system, yung ETC or electron transfer chain system, it produces 36 ATP. So indirectly, mas marami na produce na Krebs na ATP during the Krebs cycles within the mitochondrial system. Okay? So sa cellular respiration, may tatlo siyang part. May tatlong major stages in cellular respiration. One is acetyl-CoA production. So yung paggawa mo ng acetyl-CoA, which turns out to be the main component in the citric acid cycle. This citric acid cycle, you will use it to produce carbon dioxide and then to produce NADH, FADH2, which are the reduced electron carriers. Itong mga reduced electron carriers mo will be the participant naman sa electron transfer and oxidative phosphorylation. Sila yung mga kailangang component para mag-produce ng energy within this system. Okay? At yun na yung stage 3. These reduced coenzymes from citric acid cycle give their electrons to oxygen, forming ATP in the process. Right? So, ganun kahalaga yung sa cellular respiration, within you are using oxygen to produce energy. Right? Yung first stage natin, yung acetyl-CoA production, gumagamit ka na activated form ng acetate, which is meron kang ina-attach na CoA, yung acetic acid mo. So yung acetyl-CoA is an activated form of acetate Kaya siya kailangan activated para mag-undergo siya ng reaction. At the same time, makapenetrate siya dun sa loob ng mitochondria. C skeleton of sugars and fatty acid are converted to acetyl-CoA, entering CAC. Sa po muna yung plasmids. Okay? So we're on the first stage of the citric acid cycle. Si acetyl-CoA production. And we have, ang first goal mo for the stage 1 is to produce your acetyl-CoA. So, si acetyl-CoA, siya yung, kumbaga siya yung, in traveler's term, siya yung drop-off or siya yung stopover, parang gano'n, ng mga different components or different biomolecules, amino acid, fatty acid, or ng mga carbohydrates para mag-enter sa sa citric acid cycle. Okay? So some amino acids and fatty acids enter CAC via other intermediates. May other way din para maka-enter yung mga ibang biomolecules para makapasok sila sa citric acid cycle. Hindi natin sila i-cover for this moment kasi we are dealing with the from the glucose only. So sa mga susunod na topics, may mga may mga topics tayo specifically for amino acids and fatty acids. So we will cover it there kung paano sila nagpo-produce ng energy din. And the same time kung paano sila nagpo-produce. Okay? So sa conversion of pyruvate to acetyl-CoA, yung net reaction niya is that you just decarboxylate yung pyruvate mo. So you're from pyruvate, introduction of this coenzymes. So the first carbons of glucose is to be fully oxidized. So remember na sa ating glucose, may dalawa tayong pyruvate na nagagamit. So yung mag-e-enter sa citric acid cycle natin from glucose ay dalawang acetyl-CoA din. So ito, this is just a brief on how the pyruvate became acetyl-CoA. So you introduce CoAsh, NAD+, which is the acceptor of hydrogen, and another components or another coenzymes are TPP, lipeweight, and FAD. I think yung iba dito is very familiar na with you kasi they are very involved. Also, they're all involved also in the group glycolysis. Okay, so this component is this happens in pyruvate dehydrogenase complex. So may tatlong compartment yon that will make the pyruvate converted to acetyl-CoA to decarboxylate. So decarboxylate means nagtatanggal ka ng carboxylic ng carbon dioxide dun sa component. Okay? This is catalyzed by PDH, pyruvate dehydrogenase, and it requires 5 coenzymes. One is TPP, which is mostly of the coenzymes involved in PDH are vitamin B complex. So B1, thiamine, lipeweight, FAD, which is riboflavin, or vitamin B2, which are prosthetic groups. This is prosthetic, right? And NAD plus and CoAsh as co-substrates. So ano nangyayari? Ay, meron pa pala. This is the structure of coenzyme A. So si coenzyme A, yung mga coenzymes na ginagamit natin, they are not a permanent part of the enzymes. They are just nagpa-participate lang sila para mas mais or mas mapabilis then yung reaction within the system. They associate, fulfill a function, and dissociate. So bumabalik sila sa dati nilang form. They just participate para mas ma-activate, parang ganun, or mas mapabilis yung reaction, or kaya nag-carry sila ng specific product from the reaction. The function of coenzyme A is to accept and carry acetyl group. Like this, yung si coenzyme A natin, ang goal niya is to carry the acetyl group para mas makabilis, para naman makatransfer yung acetate mo to the, or makapasok yung acetate mo to the citric acid cycle. So ito yung tsura ni coenzyme A, meron siyang mercaptoethylamine. So dito naka-attach dito nag-a-attach yung acetate mo. So yung H dito will be removed and substituted with the starting materials. It can either be acetyl or yun sa mga susunod nating topic, yung mga fatty acids, it will be a long chain of alkane group or containing alkene. Meron siyang pantothenic acid, and lastly is the the adenosine diphosphate. So this whole structure or whole compound is your coenzyme A, which makes the compound or our acetate, specifically here the pyruvate, to be activated. So ito ay thioesters which have a high acyl group transfer potential, can donate the acyl group to different groups. Alright? Meron din tayong it has been erased. Alright. Wala siya dito. So supposedly, it should be explained ano yung lipoic acid. So, yung mga lipoic acid naman, ayan, mga polypeptide chain siya that helps also to transfer or wala siya dito. Alright. Yung, yung pyruvate mo. So, this is a component ng pyruvate dihydrogenase complex para lang makapag-produce ka nga nung acetylcholine. So, i-edit ko na lang siya later before sending it to you. And, sayang natanggal siya. Okay? So, ano pang meron kay pyruvate dehydrogenase complex? So, si PDH ay isa siyang malaking protein, and tinatawag natin siyang complex kasi it contains nga different enzymes that functions with each other. Okay? So, large enough to be seen with cryo-electron microscope. So, pwede siyang ma-view in CEM. And it contains 3 enzymes: pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. And these 3 components will be the will be the responsible protein to convert your acetate or your pyruvate to acetyl-CoA. So, it is a large complex like this one. Yan. Ito yung itsura ni pyruvate dehydrogenase, yung 3D constructed na from cryo-M. At it has 3 different parts: yellow, red, and green. And may specific nga silang gamit, specifically for the production of acetyl-CoA. So, meron kang E1, may E2, and may E3. And it has may mga sites sila for the coenzyme, especially yung si E1, meron siyang for TPP, si E3, meron siyang FAD, right? So, ano yung advantage ng multi-enzyme complex? Yung short distance between catalytic sites allows channeling of substrate from one catalytic site to another. This is actually very helpful kasi kapag magkakahiwala yung component ng production from acetate to acetyl-CoA, possible na magkaroon ng unwanted reaction si acetate before siya ma-convert, before siya pumasok dun sa next na enzyme or protein. So yung pagiging short distance between this catalyst or enzymes is a beneficial para maproteksyonan yung iyong compound. Channeling minimizes side reaction, as I have explained earlier. Regulation of activity of one subunit affects the entire complex. This is also beneficial kapag i-regulate naman yung pyruvate dehydrogenase complex. Kasi once na may area ko doon na i-halt mo, mag-introduce ka ng inhibitor, affected na yung buong system. Okay? Ayan, ito yung itsura ni PDH. Okay, so the citric acid cycle, so ito yung itsura niya. So you have 8 steps here, and it involves different type of reaction. You will be having formation of citrate, nagkakaroon ka ng isomerization, oxidation, will you be producing carbon dioxide. Then another phosphorylation that you will produce naman another uh, transfer, or you transfer phosphoryl group to another phosphoryl group acceptor. Then you undergo dehydrogenation to give reduced FADH2. Yung mga reduced form naman yan will be participant to other metabolism. Then you have hydration and dehydrogenation. And kaya siya cycle kasi yung napoproduce mong compound dito, yung endpoint niya will be the same compound na magiging participant for another reaction. So na patuloy siyang nagagamit and patuloy siyang nagko-continue unless you inhibit one of its enzyme. Okay? So isa-isahin natin yung 8 steps na yan. So sa step 1, yung formation of citrate. So una nangyayari natin is to form its citrate by reacting it with oxaloacetate. So yung acetyl-CoA mo reacted with oxaloacetate, the only reaction with carbon-carbon bond formation. So dito lang tayo ito lang yung part of the reaction of the TCA, whereas nagkakaroon ka ng C single bond C bond formation. So it uses acid-base catalysis. Yung carbonyl mo of oxaloacetate is a good electrophile which stabilizes the carbon ions, and yung methyl group mo has been converted to methylene. Right? So yung CH3 mo will be converted to methylene. Samantalang sa oxaloacetate mo, it is a good electrophile. Okay? So introduction of CoAsh then, with the use of water and release of CoAsh, you will now have your citrate. Okay? So ito yung rate limiting step ng CAC kasi this is the startup of your citric acid cycle. It is highly thermodynamically favorable or irreversible since you are in negative delta G prime knot. So you produce, this reaction is a very favorable reaction. So pa-left, tama ba? This is pa-right. Pa-right yung kanyang reaction. It can be regulated by substrate availability and product inhibition. Pag sinasabi natin product inhibition, yung mismong concentration or amount ng product mo na napoproduce, yun yung mismong signaling kung mag-stop ba or mag-continue yung production using citrate synthase. Okay? So you are using citrate synthase here as your enzyme to convert this acetyl-CoA and oxaloacetate to citrate. Okay? So yung citrate synthase natin, it is a very interesting enzyme. Oxaloacetate binds first. So may induced fit na nangyayari. Nagkakaroon ng closing. Ayan. So ito yung initial mo. Open conformation. So may yung free enzyme does not have a binding site for acetyl-CoA. So, anong nangyayari, yung oxaloacetate nyo and acetyl-CoA will be present dun sa active site ng enzyme. And after closing, nagkakaroon na ng binding within the oxaloacetate and the acetyl-CoA. So, and this is good also to protect your produced carbon anion. Okay? So, may mga ganun tayong enzymes that we will be encounter within the class that will function interestingly. Kaya naman ganun yung function nila is dahil, nandiyan na sila? Kaya naman ganun yung function nila is either to protect nga yung product mo, and also para it can either be to help din mas mapabilis yung transfer of another reaction. Okay? Step 2, after you form the citrate, your next to do is to produce or to produce isocitrate. So sa isocitrate, this is a isomerization or transfer or isomerization or nag-move ka lang nung OH group mo. Okay? So elimination of water from citrate gives a C double bond C. Ito yung ating intermediate. Cis is aconitate. And cis aconitate will be reacted with another water to form your isocitrate. Isocitrate, ba't kailangan natin siyang gawin? Si citrate, isa siyang tertiary alcohol. So, ito siya. So, meron siyang 1, 2, 3 carbon attached to it. And this is a very poor substrate para ma-oxidize. We cannot do furthermore oxidation in this part kasi tertiary alcohol na siya. So kaya required natin siya i-convert to secondary alcohol by isomerization using aconitase para available na siya ulit for oxidation. Addition of water to cis-acetate is stereospecific either to form isocitrate or citrate. So this is a reversible reaction with delta G of 13.3 kilojoules per mole. So you just remove the OH group and H group by hyd- uh, hydration or removal of water. Then you will react it with another water para magkaroon naman ng OH group doon sa 1, 2, 3, 4 sa fourth carbon. Alright? So you have now your isocitrate. So ayan, within the, meron naman dun sa enzyme mo. So this enzyme, it appears in cells nga, in cytosolic isozymes, it uses NADP plus as a cofactor. So gumagamit siya ng uh, uh, cofactor NADP plus, which is also a electron or a proton acceptor. So this is thermodynamically unfavorable or reversible dahil nga sa mababa niya or sa positively charged niya energy. So this also is uses iron sulfur center. So it does help yung water removal from citrate and subsequent addition to cis-aconitate are catalyzed by the iron sulfur center. So it helps yung further removal and addition of water. This is sensitive to oxidative stress, and the iron sulfur center acts in both substrate binding and catalysis. It also not help dun sa substrate binding, yung pag-remove and pag-attach nung water, but also as a catalysis. So ito yung enzyme mo, yung aconitase mo, and within this, meron kang base and the other residue of cysteine that binds with the iron sulfur complex, and it binds with your citrate. Afterwards, you will have now your isocitrate. Okay? So aconitase is a moonlighting enzyme. It means nagpo-function siya in other ways. Sa enzyme niya, yung enzyme na ito, hindi lang siya nagpo-function as to help yung hydrogen addition or hydrogen removal sa citrate mo. When iron is deficient within the system, aconitase loses its iron sulfur center and acquires a new role in iron homeostasis. Okay? So cytosolic aconitase is an enzyme with iron and sulfur and a regulator of protein synthesis. In human, iron levels must be regulated. So yung kapag too little, magkakaroon tayo ng anemia. Kapag sobrang dami naman ng iron mo, possible na magkaroon ka ng liver damage. So it acts as a transferrin carrier of iron in blood. It can be also transferrin receptor which receives an endocytosis of iron, and it can also be a ferritin or stores excess iron inside the cell. So hindi lang siya basta nag-act as enzyme for the conversion of citrate to isocitrate, and once nga na may deficiency in iron, it can be also this three, right? Aconitase in deficiency of iron regulates protein levels by stabilizing or destabilizing the mRNA of transferrin receptor or ferritin. Ayan. So yan lang naman yung other function ni aconitis. Alright? After you produce your isocitrate, the next goal is to form your alpha-ketoglutarate. So you will oxidize your isocitrate to form your next component. And within this reaction, nagkakaroon ka na ng release of carbon dioxide. Carbon is lost as carbon dioxide and NADH is generated or NADPH. Carbon loss of as carbon dioxide did not come from acetyl-CoA. So yung carbon dioxide that will be released within the system does not come from acetyl-CoA. Nagmumula yon dun sa oxaloacetate natin. Oxidation of the alcohol to ketone transfer a hydride to NAD+. So you, in here, within NAD+, nagkakaroon ka ng oxidation within your secondary alcohol. Kaya kailangan natin siyang i-isomerize. So yung carbon mo dito, magkakaroon yan ng double bond with O since you oxidized it. And nagkakaroon ng removal of carbon dioxide dahil nagkakaroon ka dito na movement. So yung mag-move yung electron mo here and this bond, ayan, yung C bond will be broken and mag-transfer yan to form another alkene group and mag-move yung electron to oxygen. Naka-release na yung carbon dioxide mo and since this is a unstable compound, mas prefer niya na yung oxygen will be in stable form. Mag-move yung electron mo to capture another proton to form your alpha-ketoglutarate. So yung nangyari dito, yung isocitrate is oxidized by NAD+ or NADP+. It depends kung ano yung enzyme, the hydrogenase isoenzymes, kung saan siya mangyayari. To form an intermediate oxalosuccinate, this will be decarboxylated using manganese 2+ as a helpful stabilizer of the electron within the compound. And afterwards, magkakaroon ng rearrangement of enol to form your alpha-ketoglutarate. So this happens in cytosolic isoenzyme, gumagamit ng NADP+ as cofactor. This is a very favorable reaction and can be regulated also by product inhibition or ATP. So this, nasa pangatlong compound pa lang tayo and you produce alpha-ketoglutarate. So what happens? You produce carbon dioxide and meron ka na nagkaroon ng transfer of proton. Alright? So next will be the oxidation of alpha-ketoglutarate to succinyl-CoA and carbon dioxide. So you will form another carbon dioxide with the step 4. This is the last oxidative decarboxylation. And what happens here? So yung carbon dioxide mo dun sa end part ng alpha-ketoglutarate mo will be decarboxylated and will be changed with succinyl with acetyl with CoAsh, okay, with coenzyme. So yung succinyl-CoA is another high energy thioester bond. So this succinyl-CoA para mas lalong important na ma-convert siya on having coenzyme A dahil activated form ulit siya para mas mag-undergo pa siya ng different reaction. So as you can see here, we have introduced another NAD+ acceptor. So within the reaction, you produce NADH and carbon dioxide. So these components will be another part of the another participant for production of ATP, specifically on the electron transport chain or in ATC. Okay, so you use here alpha-ketoglutarate, the hydrogenase complex, and it is also regulated by product inhibition. So as you can see, marami na tayo so far na mga thermodynamically favorable or irreversible reaction. It can give us idea din na kung saan marami tayong certain point na pwedeng ma-regulate para hindi mag-continue yung reaction. Okay? So doon lang naman tayo usually nagkakaroon ng regulation of reaction dun sa mga highly favorable reactions. Okay? So this is just a recap kung ano nangyari doon sa ating pyruvate kanina, which turns out to be acetyl-CoA. So yung origin ng carbon atoms within the production of the carbon dioxide ay hindi nanggagaling dun sa ating two pyruvates. It came from the carbon atoms of the oxaloacetate. Okay? Ayan, yung dalawang carbon natin na yan comes from the oxaloacetate. Okay? Ayan. So sa alpha-ketoglutarate, it is complex similar to pyruvate dehydrogenase. Yung pyruvate, ang kulang. This is supposedly ketoglutarate dehydrogenase. So it behaves the same with the pyruvate dehydrogenase and same components and same mechanism with the pyruvate dehydrogenase. Yan, ito yung example niya. So sa citric acid cycle, say alpha-alpha ketoglutarate, you produce same carbon dioxide, you use OA, you let an AD plus to accept one of your proton, the same with the other dehydrogenases. Right? Next will be the step 5 or conversion of the succinyl CoA to succinate. Okay? Ano nangyari dito? Meron kang production of GTP. Generation of GTP through thioester and substrate level phosphorylation. The energy of thioester allows for incorporation of inorganic phosphate into ADP or GDP to make ATP or GTP. Okay? So, ito yun. Succinyl-CoA mo. Succinyl-CoA will allow yung transfer ng inorganic phosphate to one of your phosphate group acceptor, yung GDP, to produce GTP. So, ito is one of also a energy generating compound or high energy containing compound. And then, you will have your succinate. So, ito yung structure ni succinate nyo. Okay? And you are using succinyl-CoA synthase. It produces GTP, which can be converted to ATP or ATP directly. This is slightly thermodynamically favorable. You have negative 2.9 kilojoules per mole. Okay? Next will be the oxidation. Oh no, hindi ko siya nabago. Fumaric. Wait lang, baguhin ko muna. Succinate of fumaric. Right. Here, you oxidize your succinate and you will have your fumarate. So, ang nangyayari dyan is you just remove your two hydrogen. So, you form an alkene. And remember na kapag nag-form tayo ng alkene, we are using FAD plus to be the proton acceptor. So, you will produce FADH2. This FADH2 will be another participant for the ATC para makapag-produce pa ng ATP or energy. This is bound to mitochondrial inner membrane or complex 2 in the ETC. Kaya very direct, ito naman yun nga, kaya yung FADH2 will be involved sa another ATP producing system, specifically yung ating ATC. Okay? Oxidation of the alkane to alkene requires FAD. FAD is covalently bound and gumagamit ka ng tatlong iron sulfur clusters. So this is near equilibrium or reversible and product concentration kept low to pull forward. So it can either be go here or go there depende dun sa demand ng cell. Okay, so you just convert your succinate to fumarate by removal of this hydrogen to form an alkene. And this is fumarate. Yun yung nangyari sa kanya. Next will be the hydration of fumarate to malate. Okay? This is highly stereospecific. So as you can see, you will be using dash and wedge lines. Addition of water is always trans and forms L-malate. So yun yung kailangan niya. Si L-malate lang yun nangyayari. Hindi pwedeng basta malate lang. So you will use L-malate. So here, yung fumarate mo in the introduction of the hydroxyl group from water, you will now have an attack in here, attack dito, then mamumove yung electron mo with this, in this carbon, and you will produce a carbon and ion, which is your intermediate. Then after addition of proton, within also the fumarase, you will now have your L-malate. Okay? This is a slightly favorable or thermodynamically favorable reaction since you have a negative 3.8 kilojoules per mole. OH adds to the fumarate, then H plus adds to the carbon ions. Yun yung nangyayari dun sa step 7 natin sa citric acid cycle. Cannot distinguish between inner carbons, so either it can gain OH or not. So this is a reversible reaction. And it will be either be formed or babalik siya sa fumarate. Okay. Last step will be the oxidation of malate to oxaloacetate. So ito yung final step of the cycle whereas yung malate mo will be oxidized at pag introduce ka ng NAD plus as acceptor of your hydrogen or your proton to form your oxaloacetate. It regenerates oxaloacetate for another citrate synthase. So ito yung oxaloacetate mo will be participant naman ulit for the formation of another citrate. And yung citrate na yun will undergo to another citric acid cycle para mag-generate ulit ng energy. So ito yung sinasabi natin na cycle kasi umuulit yung product ay yung product na mapoproduce will be the same starting material na magre-react with the acetyl-CoA para ma-produce or makapag-produce ng energy. This is very unfavorable or reversible. So pwede rin magbalik siya doon sa L-malate. And para ma-regulate siya, you have to keep yung concentration ng oxaloacetate at very low concentration. Okay? So that involves ito yung meron sa citric acid cycle, yung 8 steps natin from from initially is the formation of your acetyl-CoA, the activated form of acetate which will allow para maka-enter siya sa mitochondria, and then you will form your citrate with oxaloacetate. Then yung citrate mo na ma-form will be isomerized to form isocitrate to allow the easier or the mas ma-allow yung oxidation. Wait, excuse me. Jay, pengi akong hangin. Tapos buksan niyo yung bintana, kulong mo yung pusa. Alright? Then afterwards, yung isocitrate mo, will be oxidized ka. So, kailangan natin siya i-oxidize for the production of CO2 and NADH, mga proton acceptor that will be involved in production of other of energy of ATP. Then afterwards, your produced alpha ketoglutarate will be also converted to succinyl-CoA and mag-release ka ng carbon dioxide. Okay? Next step will be the formation of succinate by production naman ito ng GDP or ng GTP from GDP and inorganic phosphate. And lastly will be the number 6, next will be succinate to fumarate, another production or acceptor of proton. Involved siya sa electron transport chain. Okay? And next will be the conversion of fumarate to L-malate by introduction of water. And lastly will be the conversion of malate to oxaloacetate, which will be involved nga dun sa another cycle of citric acid cycle. Okay? So nakatapos tayo nung ating citric acid cycles from citrate, isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. Okay? So, question so far on this TCA. Questions? Wala pa naman so far. Okay? Chat ba yan? Or wala po, sir. Okay? Noted on that. Alright. So, ngayon naman is, ano yung, tapos na natin yung citric acid cycle. So, what's with it? So, ang goal natin dito sa mga metabolism and either catabolism man siya or anabolism is to evaluate yung energy na ginamit nila or energy na na-produce nila, which is a very good way to understand kung paano nag-regulate ng energy nga ang system or ang cell. So, sa net oxidation, meron tayong dalawang carbon na naging carbon dioxide is equivalent to 2 carbons of acetyl-CoA but not the exact same carbon. So, nanggagaling yung carbons na nanggagaling yung carbon atom, nanggagaling yung carbon dioxide na naproduce mo, yung carbon atoms nun ay nanggagaling sa oxaloacetate, not on acetyl-CoA. Okay? Energy captured by electron transfer to NADH and FADH2. So, yung net result mo of acetyl-CoA, this is just a 1 acetyl-CoA. So, remember na dalawang acetyl-CoA yung napaproduce mo sa glycolysis. Pero this is just 1 yung mapapresent natin. So, 2 acetyl-CoA reacts with 3 NAD plus and 1 FAD, 1 GDP, 1 inorganic phosphate, and 2 water to produce 2 carbon dioxide, 3 NADH, FADH, GDP, CoA, and 3H+. Yung mga, itong tatlo na yan is a very important signal to produce yung signal para malaman natin kung gaano karaming energy yung kailangan natin. Okay? Napaproduce natin. It generates 1 GDP which can be converted to ATP. Okay? Si GDP will be converted to ATP. Si FADH2 and NADH, yung protons nila will be involved in electron transfer. Okay? So this is the direct or indirect ATP yield of your citric acid cycle. Meron din pala dito kasama ang glucose, right? So, meron kang glucose to glucose 6-phosphate. You use 1 ATP. So, generated ka ng negative 1. Gumamit ka ng ATP. F6B to F16B biphosphate. You use another ATP. So, may utang kang ATP. But for the production ng glyceraldehyde to bisphosphate glycerate, you use 2 NADH, which is equivalent to 3 or 5 energy. So this is calculated as 2.5 ATP per NADH and 1.5 ATP per FADH2. So yun yung equivalent ni NADH and FADH2. Okay? So, biphosphate bisphosphoglycerate to phosphoglycerate, nag-produce ka ng dalawang ATP, equivalent siya sa 280. And kaya maraming energy na napoproduce kasi di ba remember marami tayong NADH na napoproduce, meron tayong tatlo, hindi apat. I know, bukod tong acetyl-CoA. Okay, so here, yung sa citric acid cycle natin in here, isocitrate or alpha-glutarate, meron tayong tatlong NADH, 1, 2, 3. And since dalawa yung pumapasok dun sa mitochondria mo, you have 6. So 6 yon times 2.5, you have already, ilan yun? 2.5 times 2, 5, 15. Agad yun. Kaya here, 1, 2, 3, 15. ATP na agad yun. Sobrang daming ATP na agad nun. And bukod pa yung FADH and yung ATP from GTP. From GTP. So, yan. So, overall, with, from glucose to oxalo acid, to produce your oxalo acid, carbon dioxide, and NADH, you will be producing 30 to 32 ATP. Labas na dito yung investment mo na negative na 2 ATP. So as you can see, it is a good investment since you are only using as an initial setup of 2 ATP, then you will be able to produce 30 to 32 ATP or energy. So sa glucose, glycolysis, then production of acetyl-CoA para makapunta ka sa citric acid cycle, you now have a very a lot of ATP na na-produce. Okay? So, citric acid cycle or CAC is an amphibolic pathway. So, meron tayong mga pathway which is straightforward or it will only produce in straight path. While meron naman na ito, yung atin, catabolism, amphibolic dahil it can either be catabolism or anabolism. Okay? So it is a precursors for many molecules. Needs to be replenished by red arrows. Ayan. So, for example, yung pyruvate natin, yung glucose natin, it will be converted to phosphoenol pyruvate at pwede siyang mag-undergo to production of different amino acid and pwede siyang mag-produce ng oxaloacetate. This oxaloacetate will be involved sa citric acid cycle. Yung pyruvate natin kanina will be converted to acetyl-CoA to produce citrate. Si citrate, it can be reacted to form fatty acids or sterols. Si alpha-ketoglutarate, it can be converted to glutamate para mag-produce ka naman ng purines or another type of amino acids. Si succinyl-CoA, it can be used to produce porphyrins or heme dahil nga dun sa kanyang iron sulfur component. Si malate, and for example, si pyruvate din, pwede siya mag-enter to citric acid cycle as malate. Si oxaloacetate, it can be used to form another type of amino acid, aspartate and asparagine, to produce naman pyrimidines. Si pyrimidines and purines are very important component as they are the nucleic acid to form your DNA or RNA. Okay? So citric acid cycle, kaya very complex ng mga natin, ng mga metabolism natin because they are connected to each other one way around. Either they will be the starting material or they will be the anabolic way para maging makapag-use ka ng component within the citric acid cycle. Okay? So, pag-aaralan natin sa mga susunod nating topics on how these purines, pyrimidines are produced, how these amino acids and fatty acids are produced. Dahil yun naman yung cover ng ating biochem. Alright? So, amphibolic means it can react as catabolist. It can be both catabolism or anabolism. Catabolism, breakdown, anabolism, joining. Okay? Ayan. So it needs to be replenished by anaplerotic reactions. Ayan. So, anaplerotic reaction must replenish the intermediates in order for the cycle and central metabolic pathway to continue. So, kailangan ma-produce ng maayos yung mga components within the citric acid cycle para mag-continue yung reactions natin. 4 carbon intermediates are formed by carboxylation of 3 carbon precursors. So, these are the different anaplerotic reactions na available sa ating metabolism. Pyruvate with bicarbonate and ATP, you produce oxaloacetate, ADP, and inorganic phosphate. Si phosphoenol pyruvate can be reacted with carbon dioxide and GTP to form oxaloacetate. Si phosphoenol pyruvate then can be reacted with bicarbonate to produce oxaloacetate. As this oxaloacetate ay important nga for our citric acid cycle. So hindi lang siya nanggagaling hindi lang siya nanggagaling from the cyclic reaction, pero may mga other resources din ng oxaloacetate. Important ngayon na ma-produce dahil ito yung initial component na kailangan para makapag-produce ka ng citrate to undergo yung citric acid cycle. The replenishing and consuming reactions are dynamically balanced. CAC intermediate is at constant concentration. Regulatory enzyme inactive in the absence of acetyl-CoA. Kapag walang acetyl-CoA na napoproduce, hindi magpoproduce yung mga anaplerotic reactions ng oxaloacetate kasi masasayang lang yung oxaloacetate. Okay? Si biotin is a carbon dioxide carrier. Ito si biotin. Vitamin B7, nire-require natin tong i-intake sa food. So, as you can see, so far, almost, I think, 3 yung vitamin B na na-introduced, which is very important component of the citric acid cycle. Or ginamit natin siya for conversion of pyruvate to acetyl-CoA. And as you can see, mahalaga yung vitamin B natin. So, kaya may mga supplements na available containing B complexes para ma-regulate or para ma-maintain yung required B vitamins sa body or sa cells para makapag-produce ng energy. So, ayun. It is a cofactor in carboxylases. Biotin is a specialized carrier of one carbon group in their most oxidized state. Right? Pyruvate carboxylate has 4 identical subunits, each carrying a molecule of biotin. Si pyruvate carboxylate, hindi ko lang na-include dito yung itsura niya, pero very efficient enzyme siya since it can carry na 4 identical biotin. It is present in many food and intestinal bacteria are able to synthesize it, hence biotin deficiency is rare. So, yan. Yung biotin deficiency is a rare phenomena dahil available nga siya sa maraming foods and yung bacteria meron sa mga bituka natin is capable of synthesizing this component. Okay? Consumption of raw eggs in large quantities leads to biotin deficiency. Excuse me. Okay po. Ayan. So, yung biotin deficiency, since egg white have the protein avidin, which binds with biotin very tightly and prevents its absorption in the intestine. Kapag, yun nga, biotin deficiency can happen pa rin, pero hindi siya yung parang mutation na wala kang biotin producer. Okay? So, ayan. Another important part ng citric acid cycle ay yung mga biological tethers that allows the flexibility of your component. Okay? This all allow the enter the cells to allow the transportation of the either proton or carbon dioxide All are covalently attached to proteins. All provide flexible arms on the enzymes to which they are covalently bound. All act as tethers that move intermediates from one active site to the next. So ito yung tatlong example of the biological tethers, lipoate, the biotin, and the uh, you call this ito, beta-mercaptoethylamine and pantothenate, which is part din to ng acetyl-CoA. And this help yung yung movement ng mga intermediates from one active site to another. Ang mga active site nito or mga active part ng ating biological tethers are the sulfate and the amine group. Okay? So yung mga reactive site niya is yan, yung sulfur disulfide, yung NH and the H. Okay? So how does the citric acid cycle regulated? Regulated at highly thermodynamically favorable and irreversible steps. As we have explained kanina, pwede tayong mag-regulate or pwede nating ma-control yung reaction within the citric acid cycle dun sa mga highly thermodynamically favorable and irreversible steps. Yung mga reactions na nag-move forward at may negatively charged na delta G not prime. Okay? So for example is the PDH or the pyruvate dehydrogenase complex. Bago pa magkaroon ka ng acetyl-CoA, pwede mo siyang ma-regulate using AMP, CoA, kapag mataas yung ATP, hindi na magko-convert yung pyruvate to acetyl-CoA. Kung walang acetyl-CoA, walang available na NADH at mataas na yung fatty acid content. Mag-undergo siya kapag may AMP, may CoA na available and NAD+, and also yung cofactor mo ng calcium 2+. Activated by substrate availability. Another example of part of the citric acid cycle that can be regulated is the citrate synthase here. Yung conversion mo ng acetyl-CoA with oxaloacetate to produce your citrate. Next will be the isocitrate to get alpha-ketoglutarate. And alpha-ketoglutarate to succinyl-CoA. So this 4 can be regulated para malaman kung kailangan pa bang mag-produce ng mga other components. And as you can see, in this reaction, you produce NADH. So sa NADH, this is a high producing ATP nga. It can be converted to 2.5 ATP. And kapag madami ng masyado yung energy na mapaproduce, pwede mong stop siya para hindi mag-continue yung reaction. Inhibited by product accumulation, mas maraming NADH, mas mabilis ma-stop yung reaction. Overall products of the pathway are NADH and ATP. Affect all regulated enzyme in the cycle. Since ito ay cycle, at initially yung mga kaya nating i-regulate ay yung mga starting reactions from reaction 1 to 3 and 4. Reactions 1, 3, 4, you can easily regulate the reaction, and yun nga, it can you can easily stop the cycle. Inhibitors na natin dito is NADH and ATP and NAD plus and AMP. Pag mataas ng NADH ay ATP, mag-stop yung reaction. Kapag yung cell naman, marami na siyang NAD plus or AMP, ibig sabihin wala ng energy na napoproduce yung cell, magko-continue yung cycle. CA2 plus in muscles activates the cycle. It signals the muscle contraction that signals na, kailangan natin ng energy. So, kailangan mag-produce or mag-undergo yung citric acid cycle. Okay. Regulation of pyruvate dehydrogenase. The PDH can be regulated in E1 either by phosphorylation or dephosphorylation. Okay, PDH kinase or PDH phosphatase are part of the mammalian PDH complex, and the kinase is activated by ATP. High ATP, phosphorylated PDH, less acetyl-CoA. Low ATP, kinase is less active, and phosphatase removes phosphatate from PDH, more acetyl-CoA. Yun lang naman yung interaction with siya. Edit ko muna to. Nabis lang pala to. Okay, we are on the last 3 slides, I think. Alright. Phosphatate is activated by insulin, phosphoenol pyruvate or AMP, and inhibited by ATP, NADH, and acetyl-CoA. I think this is the last line. Ay, no. May isa pa pala. Okay? So, citrate synthase another regulatory system that can be targeted for inhibition of the citric acid cycle is the citrate synthase, yun nga kanina. Alpha-ketoglutarate is an important branch point of amino acid metabolism. So, kapag marami rin masyadong amino acid, you can halt the formation of the alpha-ketoglutarate. Succinyl-CoA communicates flow at this branch point to the start of the cycle. Regulation of isocitrate dehydrogenate controls the citrate levels. Aconitase is reversible. Inhibition of IDH leads to accumulation of isocitrate and reverses aconitase. So since reversible yung citrate to isocitrate, depending on what is needed, yun ano yung magpo-form na or ano yung mas maging product within the citrate and isocitrate. Accumulated citrate lives in mitochondria and inhibits PFK1 in glycolysis. Ayan. So yung citrate, it leaves daw the mitochondria to inhibit yung PFK1. PFK1 or phosphofructose kinase 1 is one of the enzymes involved in glycolysis. So as you can see, they are very connected with each other. So kapag masyadong maraming citrate, mag-signal siya na, uy, wait lang, ibig sabihin marami na tayong energy na napoproduce, stop muna natin from the very beginning to the glycolysis bago pa ma-produce yung mga pyruvate and acetyl-CoA. Okay? So that's how the compounds within the metabolism interact with each other. They can be the starting material, they can be also the signaling compound to halt or to stop the cycle or the metabolism, or they can also be the products from these different starting material. Ayan, so they are very connected with each other. Kaya mapapansin nyo, or kung mariremember nyo, na yung metabolism natin, both the catabolism and anabolism is a very complex interaction system. Yung para siyang vein in the body na kung saan it flows ng maayos. Nakakatuwa within the metabolism on how these reactions are connected with each other and they can also regulate with each other. And kaya mahalaga rin na we understand nga yung effect nitong mga systems na ito, especially yung enzymes nila. Kasi kapag meron kang, dahil nga connected sila with each other, kapag may deficiency or may mutation within the enzymes or may unwanted behavior ng mga enzymes natin, it can cause unknown behavior or unknown disease, or not unknown disease. It can cause abnormalities within the system that may cause or may lead to defects or diseases. For example, kapag meron kang mutation within the citric acid cycle, it can lead to cancer. So this is a very rare naman daw yung mutation within CAC. But if you have, or if it will be encountered if someone has a mutation in the citric acid cycle, yung genetic defect in the fumarase, pwede kang magkaroon ng smooth muscle and kidney cancer. So, ganun siya kalala kapag meron kang mutation or meron kang abnormalities with the specific part of the citric acid cycle. Mutation in succinate dehydrogenase can form tumors of the adrenal glands. Both enzymes are defined as tumor suppressor genes. So, nagsusuppress pa sila ng tumor o means ano pa sila, i-induce pa sila ng tumor. IDH mutation leads to a new function of the enzyme that net result of which is development of glial cell tumors in the brain. So as you can see, dahil kahit isang enzyme lang yung maging involved or magkaroon ng issue within the cycle, drastic na yung effect niya dun sa system. Specifically on citric acid cycle, it can lead to cancer. So yun. Kaya mahalaga na ma-identify din kung anong part of, especially if you will be planning to enter medicinal chemistry, as I have said nga, parang maraming beses na you have to understand yung roles or the functions of the enzymes within the body kasi usually sila yung mga tina-target of drug. Kasi sila yung mga involved in different interaction or reaction, and deficiency of this enzyme may give us abnormalities that can result to specific diseases. Kaya yun. So, so kung mag-enter nga kayo na medicinal chemistry or you want to pursue med or medicine or ano ba bang pwede nyo i-pursue na. If you will want to pursue medicine, kasi ang chemistry naman ay pre-med course. So if you want to pursue medicine, mahalaga na maintindihan nyo yung biochemistry in terms of its functions, related enzymes, and the other components, either coenzyme o cofactor, na kailangan mong targetin if you are wanting to solve some diseases. Ayan. So I think that ends the lecture. May questions ba kayo? Short lang pala, 1 hour and 20 minutes. So dapat matatapos tayo ng maaga kanina. So forgive me guys, I overslept. Right? So this is the citric acid cycle. May question ba kayo?